No, go ahead. Go ahead. I've just started the recording. Sorry.
Ah, okay. Yes, my name is Sean Schaefer. I'm an associate professor at the University of Tennessee Institute of Agriculture. And in part of my talk, I'm going to introduce the department that I work in and some of the research that we do as a department. And then I'm going to focus a little bit more on my research. I decided to go broad rather than deep, so I hope that's useful to folks.
Thank you. And just on the behalf of the UT organizers and our faculty and colleagues at NIET, I would like to welcome Dr. Schaefer to this talk. This is our 9th webinar, and just for context, we have covered a wide range of topics. Some of them have focused on scholarly accomplishments or initiatives at UT, and some others have focused more on the administrative aspects, such as how to further develop our collaboration. And we've had a range of speakers on this, and we are happy to have you today, and I'm sure that this every webinar that we have is kind of another step towards a more sustainable collaboration between the universities. So thank you very much, and we are ready whenever you are for your talk.
Okay, thank you. I'll go ahead and start sharing. Let's see. So, oops, that's the very end. We need to go to the beginning. Sorry. Okay. So thank you, Nanad, and thank you to here at UT, David Ader and Tom Gill, who are at the Smith International Center and made me aware of this budding collaboration between our institutions. I'm happy to talk to you today, and I'm very glad that you thought I'd be a useful addition to this webinar series. I don't know what the other topics were in the webinar series, but what I'm going to present to you today is an overview of sort of the research that we do in my department, which is the Department of Biosystems Engineering and Soil Science here at UT, and how that fits into sort of a larger context of research into food, energy, and water systems. So we'll talk a little bit about the nexus of food, energy, and water systems, and then a little bit more about my research on conservation agricultural management practices and the kind of work that I do being a soil scientist, and how I study soil carbon and soil nitrogen. So very much in terms of sustainable agriculture and nutrient management would be the areas that I do some research in. So let me see if I can get my pointer working. All right. So yeah, by way of outline, I just want to briefly introduce what I mean when I say the food, energy, water nexus, and then how my department, BESS, is short for Biosystems Engineering and Soil Science, how that fits into this framework, and then the research in my lab. So I originally had some international projects examples, but I think I took those out in the interest of time. I'll have to see. I can't remember if I left them in. But what I did leave in was some discussion of my research on conservation agricultural management. So what do we mean when we talk about the food, energy, water nexus? It is basically looking at sustainable practices, not necessarily agriculture. There's not constrained to agriculture. But looking at sort of a whole systems approach. So rather than one person who is an expert in, say, water resources and hydrology, another one person may be specialized in energy, in actually researching how people use and produce energy. Or one person such as myself who look at how to build healthier soils or increase agricultural production. Rather than looking at this from just a single viewpoint, we try to take this transdisciplinary approach when we look at the food, energy, water nexus, and try to bring all of these different expertise together to address some particular questions. And so our department, the Department of Biosystems Engineering and Soil Science, is particularly well suited for this kind of research because we actually have faculty in our department who work in all of these areas. So in some sense, we don't have to go very far to find that transdisciplinary expertise that we're looking for. And so I'm sure that you've had some of the, it sounds like you've had some presentations that show the structure and administration of the Institute of Agriculture. So I guess I don't need to go into too much detail there. But if you're not aware, our department, BESS, has 29 tenure track faculty. I think we're up to 30 or 31 now. And we maintain 2 distinct disciplines with both undergraduate and graduate degree programs. So we actually have separate majors and minors for undergraduate students, separate PhD and master's programs as well in our department. And those cover biosystems engineering and environmental and soil science. And so I'm on the environmental and soil science side of this department, but I'm going to try to do my best to introduce a few of the projects that I've worked on with folks from biosystems engineering. So some of the projects that we have worked on that I think are related to this food, energy, and water nexus, I'm going to show a couple of examples. The first is research on building and assessing the function of constructed wetlands for water treatment. So here in the state of Tennessee, we've lost a lot of our naturally occurring wetlands, either to draining for agricultural purposes or to urban development. And we know that wetlands can buffer the impact of land use change. They can reduce the occurrence of flooding in other areas, but they can also help attenuate pollution. So not only can they reduce the spread or the rate at which pollutants move through the environment, they can actually remediate some pollutants. And so some of the research that goes on in our department, this, for example, was a project that we did with Dr. Andrea Ludwig, who's faculty in BESS, was looking at wetlands that she constructed on one of our research and education centers that the University of Tennessee runs. And that would be, monitoring things like water flow, so base flow through the wetland, but also the amount of total suspended solids that come into and out of a wetland, which is an indicator of sediment loads, which can be a pretty big pollutant. I don't generally show any data here, but what I helped her with was looking at microbial transformation of nutrients, so nitrogen and phosphorus in these wetlands, and how well it was dealing with those added inputs. Another example from the biosystems engineering side is we have people who are bioprocess engineers who are actually synthesizing new compounds for use in agriculture and bioenergy production as well. Here's an example of a faculty, Dr. Doug Hayes, who specializes in bio-based products. So these products are derived from biological compounds like, for example, corn litter. You can take that cellulose and lignocellulose and actually convert some of it to polylactic acid, which is a common polymer used in plastics. So he specializes in that and has looked at a variety of uses for these polymers. And one of those is another project that we both worked on. Doug Hayes, myself, and another faculty member, Dr. Jennifer DeBrun, looked at how we could use these bio-based biodegradable polymers to construct plastic films for use in agriculture. And so these biodegradable plastic mulches can be an alternative to polyethylene plastic. You can see up here one of our experimental sites on another one of our research and education centers where we tested different materials to see how they performed in the environment. And some of the research that I was involved in was looking at the soil physical and chemical properties. So infiltration, bulk density, changes in aggregate stability, pH, and nutrient and carbon availability to see if there were any changes in soil health or soil quality that were associated with the use of biodegradable materials. And then also, which I hope will become apparent later on in my talk, how microbial communities in the soil drive a lot of these changes or carbon and nitrogen interactions in the soil, which is my main focus for my research program. And then the project that I'm going to be focusing on for the rest of the talk is just some basic research in our department. I'm not the only faculty member. In fact, we have 2 more new faculty members working in this same area of soil quality or soil health and sustainability. And that's really looking at different conservation management practices. For example, I'm going to talk about reduced or zero tillage farming and the use of winter cover crops and how that impacts soil carbon and perhaps soil nitrogen content, which in turn changes soil quality and may increase agronomic productivity, or more importantly, make that productivity, that yield, more resilient year over year. And that's going to be especially important as we deal with the effects of climate change. So a little bit more about myself. I am a soil biogeochemist. I study carbon and nitrogen cycling in the soil. And if you haven't heard the word biogeochemistry, it's literally someone who focuses on, I focus on biology, chemistry, and geology to understand how energy and nutrients are used in the soil. So if I were to make sort of a logic flow diagram or a workflow diagram for how I conduct my research, we start with maybe a problem. So for many agricultural systems, be they production agriculture or grazing pasture for cattle, or even forestry. So forests used for wood products. we have some drivers that are going to impact the ability of those agroecosystems to function. And from my research, that often revolves around long-term changes in climate, so just overall global climate change. We also have seasonal climate, so droughts and floods that cause more acute stress to the environment. But there's also changes in land use. So two of the big problems in terms of land use change are the conversion of agricultural land. So a lot of agricultural land is being converted to urban or residential use. And a lot of our agricultural land, especially pasture land, is subject to exotic plant species invasion, which can significantly alter the ability of those systems to perform the functions that we would like. So I take these problems, I look at how microbial dynamics, so bacteria and fungi in the soil. I look at how the diversity, growth, and the adaptation of those bacteria and fungi to this stress or to this land management practice, how that impacts soil processes. So I'm not strictly a soil microbiologist or a soil chemist, or even a soil physicist. I actually work across those three disciplines in soil sciences. And I do that to understand how carbon and nutrients, particularly nitrogen, are being utilized and transformed in the soil, and also being transported or translocated, perhaps outside of an agroecosystem, either through greenhouse gas release, or in some cases through leaching into groundwater, or transport via surface erosion. And all of that is important in the end. We use that to try to impart some knowledge and help with the formulation of best management practices for ecosystem services. And those can be crop production or yield, nutrient management is a big focus of our applied research. And also we have benefits to carbon sequestration and soil health, but also water quality. So the overall research goals in my laboratory and several faculty in my department, if I had to boil it down to one figure, it would be this: we try to balance crop production and sustainability. And that's really the crux of agroecosystem management. We try to balance those. Let me get both my hands in here. We try to balance one or the other because there are trade-offs with sustainability and production. But we're always looking for ways to raise both of those at the same time if we can. And one of the major pushes in the US now is to promote management practices that increase or maintain soil health. I'm not going to talk too much about what soil health is. Maybe some other presentations you've seen have talked about that. I'd be happy to talk about it in our discussion as well. So, like I said, I focus on maybe emergent impacts on ecosystem services. So how we manage our agricultural land, and then how we deal with a changing climate. And really the key processes for me for understanding how to make a sustainable and productive agroecosystem, they all center around increasing soil organic matter, increasing soil carbon, and reducing the loss of nutrients from the system, particularly nitrogen from fertilizers. And the research directions that I've taken are looking at land use change and management. So not just the conversion of agriculture and pasture lands, but how those lands are managed, be they through tillage, cover cropping. For pasture lands, it's looking at different, different forage species and how those affect soil processes, and also different grazing rotation management. And I try to look at the resistance and resilience of biogeochemical cycles. So how resistant is that cycle to a disturbance, and how resilient is it? If it is impacted by a disturbance, how long does it take to recover? And as I will continue to point out, a lot of that for me centers around the smallest parts of the biological community. That's the microbial community, bacteria and fungi that drive decomposition. For a lot of us, that's sort of the linchpin of understanding these processes. So some of the land management practices that I and other folks in my department study are conservation agricultural practices. I mentioned zero tillage, winter cover crops, the use of bio-based and or biodegradable materials. So these can be plastic film mulches. We've also done some work with biochar additions. And like I said, all of these are meant to mitigate soil carbon and nitrogen losses. We can enhance sustainability. We can, you may have heard the term intensification. That allows us to increase productivity while remaining sustainable. I want to talk in particular about two management practices in agricultural, crop agricultural systems that we have really started to encourage the use of in the state of Tennessee, and are also being pushed nationwide in the US as ways to improve soil health or soil quality. And those are no tillage or zero tillage farming and the use of winter cover crops. So the state of Tennessee has been one of the leaders in the US in promoting no or zero tillage farming. So growing crops without disturbing the soil through tillage was originally utilized in the state of Tennessee to reduce surface erosion. But it also has huge impacts on increasing carbon storage or carbon sequestration in a soil. So it's really one of the more encouraged practices by not just folks in Tennessee, but nationwide. And then the use of winter cover crops is really gaining acceptance and widespread use across the US. The state of Tennessee is no exception. So these are just crops that are planted after harvest of the main crop. And because we live in Tennessee, which is in the southeast portion of the US, we have a fairly warm and mild winter, which allows these crops to actually grow during the winter. Now, they're not necessarily harvested. In fact, in order to promote nutrient retention and put additional carbon into the soil, they're often, the residue is often just left on the field. So they can be a good source of organic carbon. And if you plant a nitrogen fixing cover crop, it's also an additional source of nitrogen to the soil. And it's organic, it's in an organic form, so it's much less susceptible to loss compared to an inorganic nitrogen fertilizer. So I'm sure you've heard about our research and education centers across the state of Tennessee. We have some very nice long-term experiments. This is an example of one at the West Tennessee Research and Education Center. So on the other end of the state from where I am in Knoxville, where we have been looking at zero tillage and winter cover crops, both nitrogen fixers and non-nitrogen fixers, in a full factorial experiment that has been going since 1981. I made this slide 5 years ago, so it's been more than 36 years. It's been 40 years that this experiment has been running. This is one of the longest running experiments of its type in the country. And so we're very excited to have this resource available to us. And I've been working since I started at University of Tennessee in 2012. I've been working in this particular set of plots. And some of the research that I've done is to focus on the soil microbial control of organic matter accumulation. So that includes both carbon and nitrogen in this agricultural system. And so like I've been saying, for me, it all revolves around microbes. They are sort of the linchpin, or we say the gateway towards the stable accumulation of organic matter. So when I say stable, I mean organic matter, organic carbon and nitrogen that's going to remain in the soil. It's going to have a residence time of centuries to millennia. And that's very important when we think about nutrient management and soil health or soil carbon sequestration. It's not just enough to grow more crops, to input more carbon into the system. We really have to think about what form that carbon is in. Because, you know, if we increase production, if we double it, if we double the amount of carbon going into the soil, that's not going to do us much good if that carbon comes right back out into the atmosphere within a decade. We really haven't gained much in terms of carbon sequestration. So we try to focus on how microbial decomposition promotes the stabilization of soil carbon. And one way to think about that is you have an active cycling pool of organic matter, which includes things that turn over fairly rapidly, like things that are soluble in water, like sugars. Sugar is very soluble. Amino acids are another thing that tend to turn over very quickly. And a lot of the microbial biomass, a lot of the internal cellular constituents tend to cycle very rapidly in a soil. We can contrast that with what we call a passive pool of organic matter, which includes also microbial residues. But these are things like the cell walls of bacteria and fungi, which can remain undecomposed in a soil for centuries. Other recalcitrant compounds, these can be both plant and microbial derived. For plants, that would be like lignin, so a lot of the structural carbohydrates of a crop. It can also be waxes, lipids that are actually leaf waxes can last for a long time. You can also have organic matter that's stabilized within soil aggregates, so it's actually physically protected from microbial decomposition. And then you can have organic matter that reacts chemically with soil minerals to form organo-mineral complexes, some of which can be very stable and long-lasting in the soil. And so we look at all of these things to try to assess how management is going to influence the accumulation of carbon and nitrogen, because the management determines what crops you plant, how much fertilizer you may add, and how much those plants grow. And it's that above and below ground input from that plant that provides the carbon and nitrogen that microbes use to turn into stable soil organic matter. Just an example of one of the things that we look at is amino sugars. So this is a good form of carbon for looking at stabilization of soil organic carbon. Amino sugars are present in the cell walls of all bacteria and even some fungi. They're often present in fairly low concentrations in living biomass, but because they decompose fairly slowly, they can accumulate very quickly over time as microbes grow, reproduce, and die. So they accumulate in the soil, contribute to stable carbon and nitrogen because they contain both carbon and nitrogen. But we know very little about where they accumulate in a soil and what is their relationship with microbial activity. Because you need microbes growing and reproducing in order to produce these amino sugars. But if at the same time they're consuming these amino sugars, you may not have a net accumulation in the soil. So we're trying to understand how these materials are accumulating in the soil. And I don't want to get into too much detail with these data, but these are data showing two different aggregate size classes. So soil aggregates of larger than 2 millimeters, which we call macro aggregates, and then aggregates of less than 0.25 millimeters or 250 microns, which we call micro aggregates. And you can see that the accumulation with different management practices, you can see right away that those amino sugars are accumulating at different amounts under these different management treatments. And this is in that long-term 40-year experiment where we did this work, because in some cases it takes that long in order to see these kind of patterns developing. But that's what we need to see. If we're trying to look at long-term carbon accumulation, carbon sequestration, we need long-term experiments. Just to make things more complicated, here's an example of how we took all that data, fed it into what's called a structural equation model, which is just a multivariate analysis that tells us sort of what are the most important drivers of amino sugar accumulation. And it turns out that soil structure, particularly the amount of microaggregates, has a strong positive impact on amino sugar accumulation, but also microbial activity. So the more microbes you have and the more they are active, that leads to more accumulation of this stable organic matter in a soil. You also have some things that are not good for this stable carbon accumulation. Those can be tillage. So you can see this lighter colored arrow is a negative correlation. So increased tillage tends to decrease the accumulation of amino sugars. And also, for some reason, the use of a wheat cover crop, so a non-nitrogen fixing cover crop, doesn't really do much to promote stable organic matter accumulation in terms of amino sugars. And somebody may ask, well, how much are amino sugars present in the soil? How much of the soil organic matter do they constitute? And the answer is we don't fully know. In some cases, it can be maybe up to 20 or 30% of what we would consider stable organic matter is in amino sugars, which is a staggering amount when you think about it. But it's really highly variable with an ecosystem. And because amino sugars can be very difficult to measure, there's not a lot of data to tell us what proportion of soil carbon they actually constitute. I'm gonna try to wrap this up now by talking a little bit about nutrients or nitrogen in the soil. I talked about carbon. Now I'm going to talk a little bit about nitrogen. We have this same plant, microbes, soil organic matter cycle that drives carbon accumulation, can also drive nitrogen accumulation. And a lot of our research, not just in my lab, but everywhere in the US, revolves around excessive nitrogen inputs. And the reason we look at that is because if you want to just look at it in its most basic problem, more nitrogen or excess fertilizer, the more nitrogen you add, the more you may lose in terms of greenhouse gases. So the big one is nitrous oxide. And so here's some data from that same field experiment in West Tennessee that shows a couple of things. First, that you can apply zero nitrogen to zero nitrogen inorganic fertilizer to the system. And if you use a nitrogen fixing cover crop, in this case vetch, you can maintain the same yield. This is for cotton. You can maintain the same yield as you would if you had no cover crop and 60 pounds per acre fertilizer addition. And this blue bar here represents the conventional management that farmers have used for years. No cover crop, 60 pounds of inorganic nitrogen fertilizer per year. But we can get that same yield with no fertilizer input and the use of a nitrogen fixing cover crop. The other benefit of potentially switching to, well, I'm sorry, this is also a no-till system as well. So no-till, no fertilizer, same yield as conventional tillage and fertilizer. This may also lead to lower greenhouse gas fluxes. So if we look here and we look at the no-till, zero nitrogen addition, you can see this black bar here is actually smaller than this yellow bar. So this is the conventional management practice. This is the full conservation management practice. And you can see when scaled for yield, greenhouse gas emissions are actually, or may actually be lower with this conservation management practice. So my lab is also looking at some of the reasons for how this is possible. And it has to do with the microbial drivers of the soil nitrogen cycle. And without getting into too much detail, I just want to show you that a lot of that research revolves around actually isolating and extracting and quantifying the genes that are responsible for transforming nitrogen in the soil. So those can be nitrogen fixation, genes that code for nitrogen fixing enzymes, as well as genes that code for ammonium oxidation and then nitrate reduction. So nitrification and denitrification in the soil. And this is one of our ongoing research topics. Paper just came out this year in EMsphere from my group looking at this. And just to wrap it up, like I said, we try to study this in terms of climate change. So not just the chronic climate change due to global warming and changing climate patterns, but also the stresses due to extreme events. So acute stresses due to floods, drought, etc. And like I said, how can we tell if our ecosystems are resilient? Do they bounce back when they're disturbed, or how resistant are they? Adaptable are they to these chronic and acute stresses? And so that work is really continuously ongoing. Again, not just in my lab, but other labs, looking at how microbial communities are changing with simulated drought and simulated heavy precipitation. Because in many cases, we can't do these types of field experiments easily because it's kind of hard to wait for an extreme drought or an extreme flood to occur on your field site. So a lot of this is lab-based research. Just in summary, I just want to say that for me and my research, sustainability, one aspect of sustainability lies in biogeochemistry. That's why I study soil biogeochemistry. And as a department, I say we study the food, energy, and water system nexus, and it's not a true food, energy, and water solution until we take a transdisciplinary approach. And I hope that that some of you, I don't know what everyone's expertise is, but I'm guessing that you also have some of this transdisciplinary expertise, which is where we can actually, I think, most effectively work together to solve some of these sustainability problems. So I'm sorry if that took more time than I thought, but I'm open to discussion and questions. Thank you for listening.
Schaefer, thank you. That was a very informative talk, and I'm happy that you kind of went started with the broad scope and then came down to your specific topics. Nurullah, do you want to summarize for your colleagues and say a few words before we take questions?
Okay, yeah, thank you very much. Thank you very much, Dr. Schaefer, for your very useful and informative presentation. As I said at the beginning, that unfortunately some of our very important colleagues are not participating here, who have you had very high interest in for question and answers at this very much point. There we have collected the questions and answers. We'll start. Questions and answers. Here we are. Some are participating from a gardening department, one from a chemical technology department, one from fiber processing industry department, and also relative departments here. Can we then go to a question and answer section then, Ninat?
Please.
Hurmatli qatnashchilar, agar savollaring bo'lsa, qishloq xo'jaligi bo'yicha hozir berishinglar mumkin. And also, at the moment also, I'm in touch with my colleagues who are participating in another meeting also, and also they are giving their questions in through SMS also as well, that they are being participated. Our colleague is interested in if there's an opportunity in cooperating in an element called hydrogel. If you're aware of that, it's a water reserving technology that if you use hydrogel to the soil, water will be reserved. So it can be used in places where water is very scarce water. So it can preserve the water for a longer time than normal.
Did you say hydrogen?
Hydrogel, yes.
Hydrogel.
Hydrogel, yes.
So I don't know. I really don't know anything about the hydrogel, and if anybody in our department is studying this particular technology, but some people who might, we have not in the Institute of Agriculture, but in the College of Engineering at University of Tennessee, we have the Department of Civil and Environmental Engineering, and there may be faculty there who are working on this problem or technology application.
And this is a kind of element that, according to them, it gives it keeps the soil damp for a longer time.
Mm-hmm.
So I'm not a specialist in agriculture, but this is what they worked out. Hydrogel.
It sounds like it's a hygroscopic compound that just retains water. There actually is a natural analog to that. Bacteria form biofilms in a soil, and sometimes in response to drought stress, bacteria will exude polysaccharides. And these polysaccharides are also hygroscopic, which means they attract and retain water. And it's a way for bacteria to remain hydrated in a soil for a longer period in order to, they're not trying to survive, they're trying to avoid a drought by surrounding themselves with a moisture layer of these extracellular polysaccharides, we call them. So that's the closest I can come to hydrogel.
I don't know the content of the hydrogel myself, as I'm not a specialist, but I believe, I don't know, maybe it can be connected to this one as well if they worked on that. I don't know how natural it is, if it's artificial, if it's a natural amount.
Mm-hmm.
I'll give the message to them anyway that there's a natural also solution analog is available, which is bacterial, yes, polysaccharides, biofilms, right? Okay, thank you very much. And one more question is there that our concern, our region is very much, our institute is very much specialized in cotton processing. And cotton processing, that's why the question relates to cotton also is there about, so there's a soil nutrient management. Is it different from crop to crop, or is it all the same for all crops? For example, what management can be used effectively, for example, to grow the cotton, for example, more effectively, could be our interest.
Well, that's an excellent question. And a lot of the work that's been done at the Institute of Agriculture is to actually determine how much, what fertilizer rate would be the most appropriate, not only for a given crop, but for different areas. So the state of Tennessee is actually fairly large. We have 3 distinct geographic areas, and we will have different fertilizer rate recommendations. We'll have different recommendations for the same crop in all of these different areas. Mm-hmm. So a good example of that is for phosphorus. So we have soils in West Tennessee that are very poor in phosphorus, but soils in the middle and eastern part of the state that are very rich in phosphorus. So when you're growing any crop, be it corn or soybeans or cotton, which are our three major crops in the state, you're going to have different recommended rates of phosphorus application. So that's what we try to do. So the answer is, not only is it different, not only is nutrient management different for different crops, it's different depending upon the soils and the climate that those crops are growing in as well.
Mm-hmm. Yeah, okay, thank you very much. That's very, very clear, very understandable. So it's not per crop, it's not depending on the crop, it's depending on the area.
Mm-hmm. And we're also looking at ways to reduce that fertilizer input. So nitrogen fixing cover crops are one of our major efforts to try to do that. You can actually reduce the need for inorganic fertilizer by proper nutrient management.
Thank you very much.
Hello.
Yes,
I
please.
have a question. Yes. Good afternoon.
Yes, please.
Yeah. Pauzbekskим говорит.
O'zbekcha gapiraylik, our gardening department.
Tarjima qilib berasiz. Ularni institutida gulchilik sohasi bo'yicha qanaqa ilmiy tadqiqotlar o'tkaziladi? Ular bilan aloqaga chiqsak bo'ladimi? Masalan, in vitro usulda, boshqa usullarda shu gulchilik sohasi, umuman olganda gulchilik sohasida nima ishlar qilinadi, shu bo'yicha gapirsalar yaxshi bo'lardi, so'rasak. Thank you.
Oh, Soraya Misirwe is the head of. That's why her question, her concern is a little bit out of the area. She's interested in if Tennessee, because you're from agriculture background, Tennessee has flowering specialists as well that we can cooperate with. If you have a knowledge about the.
Yes, yes, in the plant science department at the Institute of Agriculture, we have several faculty working on horticulture applications. So yes, there should be at least one person, if not 3 or 4, working on flowering.
Multiply in vitro, in vivo methods of multiplications, they work.
I don't know. Yeah, I don't know the specific applications that they're working on.
Okay,
That's out of my area.
Yes, yes, thank you very much.
Oya tushundingizda nima bor ekan unda?
Tushundim.
O'simliklar fani bo'yicha nimasi bor ekan?
Yes, I get it.
Okay.
Thank you very much.
Yana tuproqqa oid savollar bo'ladigan bo'lsa, marhamat. Savollaring bersinlaring. Vaqtimiz hali bor.
Ularda masalan bizda balbanitet deydida Nurullo aka. Ularda balbanitetini tuprog'i qanoq aniqlanar ekan? Bu bo'yicha gapirmadilarda. Tuprog'i balbanitet deydi. Angliska yozigi bilmayman.
Hop.
Shu balbanitetni ularda qanoq aniqlar ekan? Qanaqa metodlari bor ekan mana shu tuproqni tarkibini aniqlash bo'yicha? Masalan, makro mikro elementlari, yana другой boshqa elementlarni qanaqa usullar bilan aniqlanar ekan? Oxirgi yangi usullar bo'yicha.
Her question is, what are the modern updated and available methods of clarifying the soil content?
Did you say model? Like computer models?
Any model which determines the content of the soil, which the contents, all the contents, which the soil consists of.
Do you mean like analytical methods? How to determine?
Yes, analytical. Yes.
Analytical.
How do you analyze the soil content? I think that is the question.
Okay. Yeah, we use a wide range of methods because in my lab, not only are we looking at the bulk, so the total amount of carbon and nitrogen in the soil, but we're also trying to partition that soil carbon and nitrogen into different forms. So organic and inorganic forms in the soil. I gave an example of amino sugars as one form of carbon and nitrogen in the soil. To provide more detail, if for bulk, for total soil carbon and total soil nitrogen, the most common method we use is dry combustion. So we take a soil sample, and we combust it, convert all of the carbon to CO2 and all of the nitrogen to nitrogen gas. And then we analyze the concentration of each of those to determine total carbon and nitrogen content. There are lots of other ways to look at soil carbon and nitrogen. There's older, more intensive chemical methods that you can use. I think a common one that a lot of people used to use that we no longer use is the Walkley-Black method for measuring total carbon in a soil. But the other methods we use, most of these are, we use a series of wet chemical extraction techniques and physical fractionation of soil. So sieving a soil, suspending that soil in different liquids of different densities to get different fractions of carbon and nitrogen in the soil. What else do? We look at things like how much carbon and nitrogen is left after we react it. So if we want to look at long-lived or very stable carbon in the soil, we'll take a soil sample and we'll actually try to remove all of the active components. So we may use in my lab, we've used like hydrofluoric acid or hydrogen peroxide to oxidize or reduce most of the available organic carbon and nitrogen to look at what's left, which is like that mineral bound carbon and nitrogen that I talked about. I'm sorry, it's not a very specific answer because we use so many different methods.
Yes, thank you very much, thank you. And Nurullo aka, soil fungi haqida ham gapirsizlarchi, soil fungi, ana tuproq zamburug'lari qandoq aniqlashar ekan? Bu haqida gapirmadilar-da.
Soil fungi.
Yes.
Yeah. I think she's interested about how to determine soil fungi or fungis.
Yeah, fungi or fungi. That's, yeah, either one of those is fine.
Yeah.
The way we've been doing it in my lab is actually extracting the DNA from the soil and using a primer to actually, a primer that's specific to soil fungi in order to amplify only the fungal DNA in a soil. And so I don't know if anybody's familiar with the use of 16S ribosomal RNA for bacteria. For amplifying fungal DNA, we use the ITS region of a fungal genome. There are other ways to extract fungi. There's at least one other person on the University of Tennessee campus. She's not in the Institute of Agriculture, but she works extensively on soil fungi. And in her lab, they have methods for physically separating and counting fungal hyphae. So they sieve the hyphae out of the soil and actually quantify them that way. They also have methods for extracting carbon and nitrogen compounds from fungal biomass. So that would be chitin or chitosan, or sorry, just chitin. And also glomalin.
Yes, thank you.
Mm-hmm.
Okay, I see. Thank you very much.
Thank you very much for the question as well. If there are other questions, hurmatli qatnashchilar.
Nurullo, if there are no other questions, we can wrap up.
Is the okay? So time is already yeah, almost up. Okay. Yes, please.
Thank you, Nurullo. Thank you once again for coordinating and reaching out to your colleagues. We steadily had an increase in the participation as the webinar went on, so I appreciate that. And Dr. Schaefer, thank you once again for your time.
Yes, I'm very happy to get to meet all of you and talk about the research that we're doing. I would be happy to talk with anyone who has any more questions or is interested in a research collaboration. Please don't hesitate to contact me. If I can't help you, maybe I can put you in contact with someone at University of Tennessee who can. I'll do my best. Ninad has my email. And yeah, just please give it to people if they want to have any questions at all.
Nurullo aka, ma'ruzalar uchun katta rahmat. Juda chiroyli chiqibdi ma'ruzalar. Shunaqa ma'ruzalarni ko'p o'tkazib tursa yaxshi bo'lardi. Shuni deb tarjima qiling. Katta rahmat deng.
Yes, she's thanking you very much for the presentation. Presentation was very greatly designed. We are very glad to see that, and we hope to collaborate further. And wish you good luck in your future activities. And thank you very much on behalf of our participants from NIET side. Also, I would like to thank you for your time and for your efforts to prepare the presentation and share with us today. Thank you, Ninad, for organizing it, for making it possible for us.
Thank you for inviting me.
Doctor.
Yes,
Thank you. Have a good day and a good evening, everyone. Thanks.
Hope to see you again.
Next.
Okay.
Bye.
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 Bye.